When we talk about Alzheimer's, our minds usually jump straight to those sticky plaques gumming up the brain. But what if a huge piece of the puzzle isn't about what's being added, but about what's being torn down from the very structure of our brain cells? We're going to dig into that right now.
For decades, the story of Alzheimer's has had one clear villain, a protein called amyloid-beta, the stuff that makes up those famous plaques. But more and more, research is pointing in other directions, suggesting that the plaque might just be a symptom, not the actual cause, which leads us to a really fascinating question.
So let's meet our new suspect, a gene called CD2AP. Now, we've known for a while that certain versions of this gene bump up the risk for late-onset Alzheimer's. The problem is, how it does that has been a total mystery. We knew it was at the scene of the crime, so to speak, but we had no idea what its role was. Until now. Okay, so in any good investigation, the first step is to figure out where your suspect operates. If we can pinpoint where this CD2AP protein actually hangs out in the brain, we can start to piece together what it's doing. And this investigation takes us right to the heart of where learning and memory happen. These things called dendritic spines. They're these incredibly tiny little bumps on our neurons that act as the receiving end for signals. You can think of them as the microscopic listening posts of the brain. This is our crime scene. So how do we even know CD2AP is here? Well, this slide shows some of the key lab work. Researchers basically took brain cells, broke them into their component parts, and sorted the proteins. It's a technique called a Western blot. And you can see that CD2AP shows up in the same group as a protein called PSD95. And PSD95 is a well-known marker for the receiving side of a synapse. And just as important, it's missing from the group with the sending side markers. So the evidence is crystal clear. CD2AP is working on the listening end of that conversation between neurons. And it's not just hanging around there. It's really concentrated. The researchers figured out that 34% of all the CD2AP in a dendrite is packed specifically into these tiny spines. Now that is way more than you'd expect if it were just floating around randomly. It tells us it has a very specific job to do right there at the synapse. And this amazing 3D image gives us its exact location. This is like a high-tech forensic photo. We're looking inside a single spine. The main receiving machinery, that PSD95 protein, is right there in magenta. But look at our suspect, CD2AP, in cyan. It's not in the center. It's positioned off to the side in an area that's known for managing cellular cargo and maintaining the spine's internal structure. It's like finding a suspect not at the center of the action, but standing right next to the control panel that makes everything run. Okay, so we know where it is. Now for the classic detective move. Let's see what happens when we take our suspect out of the picture. What are the consequences when you get rid of CD2AP? And the results are, well, they're visually stunning and not in a good way. On the left, you've got a healthy dendrite, and you can see it's just covered with all those essential spines. On the right, after CD2AP has been removed, it's a disaster. The spines have just withered away. The numbers behind that image are just brutal. A 55 to 60% drop in the number of spines. And the ones that do manage to survive are 30% smaller. And since spines are where connections are made, this leads to a massive loss, up to 41% of the synapses that let our neurons fire and communicate. The entire communication network is being crippled. Now hold on. A sharp neuroscientist watching this would ask a really important question right about now. We happen to know that when you lose CD2AP, it can actually cause an increase in the production of that old villain, amyloid-beta. So is all this destruction just a side effect of having more of that plaque-forming stuff around? To find out, the researchers set up a really clever experiment. They got rid of the CD2AP just like before, but this time they also used drugs to completely shut down the production of amyloid-beta. The logic is simple. If A-beta was the only culprit, then blocking it should completely fix the spine problem. And here is the big reveal. Blocking amyloid-beta, it only brought back a small fraction of the lost spines. Most of the damage was still there. This is basically the smoking gun. It tells us that while AB might be an accomplice, CD2AP is absolutely capable of destroying synapses all on its own, through a totally different mechanism. So the plot thickens, and it gets even weirder when we look at a specific rare mutation in the CD2AP gene, one called K633R, that's strongly linked to Alzheimer's. You'd probably expect it to do the same thing, but worse, right? Well, get this. It completely flips the script. Just look at this chart. When the researchers added extra healthy CD2AP to neurons, the number of spines went up, which kind of makes sense.
But when they put in the Alzheimer's-linked mutant version, the spine density went through the roof, a huge jump over the control. So your first thought is probably, hey, more spines, that's gotta be great, right? More connections, more memory power. It seems like it should be a good thing. But in biology, especially in the brain, more isn't always better. And here is the major catch. These extra spines, they're duds. See, healthy spines are supposed to be dynamic, constantly moving and changing shape. But these new ones are less motile, almost frozen. And even though there are more of them, they aren't forming more working synapses. In fact, the overall synaptic activity gets weaker. It's a classic case of quantity over quality, and the result is a network that doesn't work right. So now we've got a real paradox. Too little CD2AP and the spines disappear, a mutated version of CD2AP, and you get too many faulty ones. To understand what's really going on, we need to look at the mechanism, how CD2AP actually does its job at the most basic level. And the answer, it turns out, lies with a protein called F-actin. The best way to think of F-actin is as the spine's internal scaffolding, but it's not like the rigid scaffolding on a building. It's incredibly dynamic, constantly being built up and torn down. And that dynamic nature is what allows spines to change their shape, to grow, and to form new connections. It's essential for learning. So how do you measure how dynamic the scaffolding is? Scientists use this really cool technique. First, they make the actin glow. Then, they use a tiny, precise laser to bleach a spot, making it go dark. After that, they just time how long it takes for new, glowing actin to move in and repair that dark spot. A faster repair time means the scaffolding is more dynamic, more healthy. And the timing here tells the entire story. In the control cells, the scaffolding repairs itself in about 6.5 seconds. Adding more of the healthy CD2AP slows that down a bit, to 9.5 seconds, which suggests its normal job is to stabilize the structure. But with that K633R mutant, the recovery slows down even more. The scaffolding becomes too stable, too rigid. It can't remodel itself properly. The researchers put it perfectly in their paper. This Alzheimer's mutation leads to faulty F-actin dynamics. The scaffolding just gets too stiff. Sure, it can build new structures, those extra spines we saw, but it can't adapt or change or function the way it's supposed to, and that ultimately breaks the synapse. So the big takeaway here is that CD2AP is like a master architect for our synaptic scaffolding. If you lose it, the whole structure collapses. If you have a mutated version, the structure becomes rigid and useless. And this tells us something profound. Alzheimer's risk isn't just about bad brain chemistry, like plaques. It's also about bad physics, the literal structural integrity of our neurons. And this just opens up a whole new way to think about treatments. Instead of just trying to clear out protein junk from the brain, what if we could develop therapies that actually target and repair this delicate internal scaffolding? Could reinforcing the physical structure of our synapses be the key to protecting our memories? It's a fascinating thought.